Hi guys, Jacob Pushy here, and today I'm going to be walking you guys through the Week 4 Advanced Concept presentation, which asks me to walk you through, walk you through the process for how nucleotides and ribose are made, and show how these come together to make ATP used for DNA replication. And then walk the class through ATP production and regeneration, or ATP synthase. Alright. So, synthesis of nucleotides is, so nucleotides can be synthesized by many different means, both in vitro and in vivo. So in vivo, protecting groups may be used during laboratory production of nucleotides. A purified nucleoside is protected to create a phosphoramidite, which can then be used to obtain analogs not found in nature, and then to synthesize an oligonucleotide. And I'm going to be at the end of this presentation, so when I explain everything, I'll then show you guys my poster. But for now, I'm just going to be talking about the processes. So, in vivo, nucleotides can be synthesized de novo, or recycled, through salvage pathways. The components used in de novo nucleotide synthesis are derived from biosynthetic precursors of carbohydrates and amino acid metabolism, and from ammonia and carbon dioxide in alternative methods. The liver is the major organ of de novo synthesis of all four nucleotides. De novo synthesis of pyrimidines and purines follows two different pathways. Pyrimidines are synthesized first from aspartate and carbomonophosphate in the cytoplasm to the common precursor ring structure, orotic acid, onto which a phosphorylated ribosyl unit is covalently linked. And then so purines, however, are first synthesized from the sugar template onto which the ring synthesis occurs. For reference, the syntheses of the purine and pyrimidine nucleotides are carried out by several enzymes in the cytoplasm of the cell, not within a specific organelle. So the nucleotides undergo breakdown such that useful parts can be reused in the synthesis reactions to create new nucleotides. And then next, I'm going to be explaining the synthesis of ribose. So ribose is a type of sugar normally made in the body from glucose. Ribose plays an important role in the synthesis of RNA and DNA and the energy-containing substance ATP. So the pentose phosphate pathway, otherwise known as the phosphogluconate pathway, and the hexose monophosphate shunt, is a metabolic pathway parallel to glycolysis. It generates NADPH and pentoses, as well as the ribose 5-phosphate, which is the last one, a precursor for the synthesis of nucleotides. So, glycolysis begins with glucose metabolism in all cells to produce two molecules of pyruvate, and it occurs outside of the mitochondria, usually in the cytoplasm. Cellular respiration uses oxygen from the environment and converts each pyruvate to 3 molecules of carbon dioxide while trapping the energy released in this process in ATP. And most ATP produced by aerobic cellular respiration is made by oxidative phosphorylation, and this works by the energy released in the consumption of pyruvate being used to create a chemiosmotic potential by pumping protons across the membrane. All right. An ATP is a nucleotide that consists of 3 main structures: the nitrogenous base, adenine, the sugar, the ribose, and a chain of 3 phosphate groups bound to ribose, as we know. The phosphate tail of ATP is actual power source, which the cell traps. And ATP synthase, as ASE indicates, is an enzyme that creates the energy storage molecule adenosine triphosphate, ATP. So ATP, most commonly used energy currency of cells for all organisms, as we have discussed, is formed from adenosine diphosphate, or ADP, and inorganic phosphate, PI, as we know. The overall reaction catalyzed by ATP synthase is ADP plus PI plus H plus out yields ATP plus H2O plus H plus in. And then lastly, ATP is a nucleoside triphosphate, where a high energy bond attaches the third phosphate group to the molecule, making it the triphosphate. This bond is highly unstable, and when it is hydrolyzed, it releases a substantial amount of free energy, which is approximately 7 kilocalories per mole. In addition to providing energy, ATP has other essential roles within the cell. It is one of the 4 nucleotides required for the synthesis of DNA and RNA. It regulates certain biochemical pathways in mammals, and it is released from damaged cells to elicit a pain response. And in photosynthetic organisms, it drives carbon fixation. Nonetheless, as it is unstable and is used for almost every conceivable process, each cell in the body must constantly produce ATP to supply needs. So in total, an organism's requirement for ATP is substantial. The average human generates around 100 kilograms of ATP per day. ATP synthase is the prime producer of ATP cells, catalyzing the combination of ADP with inorganic phosphate to make ATP. Alright, so now I'm going to show you guys my poster. I don't know. Yeah, there it is. So here we begin at the top. And just as kind of I explained in the processes here. And we all have the same poster, hopefully, so we all kind of know what that entails. All right. Thanks, guys.